Hello, students, and welcome to the today's online session. Hope my voice is audible to you, and the screen I have shared with you is clearly visible to you all. If it is, please write in the chat box so that I can get an idea that I am clearly audible to you and my screen is clearly visible to you all. Yes. So, hello, students. So, the topic of today's online session or webinar is liver function test. So, in this class, we'll be talking about the liver, what is liver, what its function is, how it performs its vital functions in the body, what are the disorders that are related to it, and the diagnostic tests that are performed in the laboratory for the diagnosis of liver, that if the liver is performing well in the body or if there is any kind of disease or disorder that is associated with the liver. So, without delay, let's start today's session. So, the objective is to enumerate functions of the liver, first of all, that what functions of the liver are. Then we will be discussing about the biochemical tests which are done to assess the functions of the liver. Thereby, we'll be talking about the biochemical basis of the liver function test, and then we'll be discussing about the biochemical alteration in the patients with the jaundice.
There's a factory inside you that weighs about 1.4 kilograms and runs for 24 hours a day. This is your liver, the heaviest organ in your body and one of the most crucial. This industrious structure simultaneously acts as a storehouse, a manufacturing hub, and a processing plant. And each of these functions involves so many important subtasks that without the liver, our bodies would simply stop working. One of the liver's main functions is to filter the body's blood, which it receives in regular shipments from two sources. The hepatic artery delivers blood from the heart, while the hepatic portal vein brings it from the intestine. This double delivery fills the liver with nutrients that it then sorts, processes, and stores with the help of thousands of tiny internal processing plants known as lobules. Both blood flows also deliver the oxygen that the liver needs to function. The blood that is received from the intestine contains carbohydrates, fats, and vitamins and other nutrients dissolved in it from the food you've consumed. These must be processed in different ways. In the case of carbohydrates, the liver breaks them down and converts them into sugars for the body to use as energy when the filtered blood is sent back out. Sometimes the body has leftovers of nutrients that it doesn't immediately require. When that happens, the liver holds some back and stacks them in its storage facility. This facility works like a pantry for future cases when the body might be in need of nutrients.
So, first of all, what is liver? There is a question that what is liver? So, liver, if we talk about liver, it is a versatile organ, we can say, which is the central organ of the body metabolism and independently involved in many biochemical functions. So, liver performs several diversified functions in the body. It is the largest solid organ. It is located in the right upper quadrant. It is large. It has a large reserve capacity, and the cells of the liver has the highest capability of the regeneration. Let's discuss some of the functions of the liver that are performed by the liver itself. So, first of all, there is a metabolic function. So, liver is the key organ and the principal site where the metabolism of carbohydrates, lipids, proteins, minerals, and vitamins takes place. Second function is the secretory function. So, liver is responsible for the formation and secretion of bile in the intestine. Bile pigment bilirubin formed from heme catabolism is conjugated in the liver cells and is secreted in the bile. Cholesterol and bile salts are also secreted in the bile into the intestine. So it is the secretory function of the liver. Next function is of the liver is the excretory function. So exogenous dyes such as BSP, which is bromosulfothylene, and rose Bengal dye are excreted through the liver cells. Next is the detoxification and the protective function. So, by the liver, ammonia is detoxified to urea. Liver cells are capable of detoxifying drugs, hormones and convert them into less toxic substances for excretion. So, for example, the Kupffer cells of the liver perform phagocytosis to eliminate the foreign compounds from the body. Next function of the liver is in the storage function. So liver also acts as reserve capacity for the storage of different compounds. One such example is that it stores glycogen, trace mineral, iron, and vitamin A, D, and B12 in it. Next function of the liver is the hematological function. So, liver participate in the formation of blood, particularly in the embryo and adults in the abnormal states also. So, in synthesis of plasma proteins and blood clotting factors and the destruction of erythrocytes is also performed by the liver sometimes. So, these are the functions of the liver. Hope it is clear to you. So there are let's now talk about the some examples of the liver dysfunction. So there is hepatocellular diseases that can be occur if the liver is not performing well, or if there is some sort of damage to the liver. It can be due to viral hepatitis or acute liver disease. Next is cholestatic disease. So cholestatic disease may be caused by the intra and extra hepatic obstruction. It is usually caused by the adhesion of fat to the liver. Next one is the cirrhosis. Next is cancer of the liver. It can be primary or secondary. Next is the fatty liver, which is caused due to the aggregation of fat on the liver. Next is some kind of genetic disorders may also be there in the liver, such as first one is the hemochromatosis, in which there is increased iron storage in the liver, and second one is the Wilson's disease. So these are the examples of the genetic diseases that may be associated with the dysfunctioning of the liver. This is clear to you. So how we will be diagnosing the liver dysfunction? So it majorly depends on the combination of different tests. So numerous laboratory investigations have been proposed in the assessment of laboratory liver diseases, which are also called as the standard liver function test, that will help to detect the abnormality and the extent of the liver damage. So the basis of the diagnosis depends on the combination of patient history, its physical examination, laboratory testing, biopsy, and some of the imaging studies can also be deployed, such as ultrasound, CT, or MRI scan. Now, talking about the liver function test, so there are different types of liver function tests that are performed in the laboratory that are used, first of all, to detect the presence of any kind of disease in the liver. Second one, to distinguish or to classify among different types of liver disorders, that which kind of liver disorder is there in the is present in the person that has been diagnosed. Next is to gauge the extent of known liver damage. The liver function tests are also performed to know whether the liver has been damaged more, or it has been damaged less. So to know the extent or severity of the damage of the liver, the liver function tests are performed. Next, to follow the response to treatment, or we can say the to know about the prognosis of the treatment, that how well the treatment that has been given to the patient is performing in the patient's body. So to know the response of the liver to the treatment, the liver function tests are also performed. So, what are the shortcomings now of the liver function test? So, the shortcomings can be normal in a patient with serious liver disease, and abnormal in a patient with disease that do not affect the liver. So basically, we can say, or we can conclude by this line, that the shortcoming is that the false positive results or false negative results can be there, which may lead to the erroneous test results in the laboratory. Next, it rarely suggests a specific diagnosis, rather suggests a general category of the liver disease. So we can say that the LFTs that are performed, they do not specify a specific diagnosis until and and unless we are performing the special test. All right. So the diagnosis of the liver function test generally gives an idea of the category or the classification of the liver disease that is present in the particular patient. So we can say it directs the evaluation process. All right. So there are standard liver function tests which are classified on different bases. First of them is test based on metabolism. Then there are different classification. Then there is a test based on the detoxification and protective functions of the liver. We'll be talking about all these. Let's first note some of the important points that are there which are associated with the liver function test. So first of all, liver is a place where thousands of biochemical functions takes place, most of which cannot be measured, but some of it can be measured. So enzymes do not measure liver function at all, detect damage or interference with the bile flow. So the enzymes, the increase or decrease in the liver enzymes do not measure the liver function, that how well the liver is functioning. And perhaps it detects the damage that is present with the liver and the interference of the bile in the liver. Next is interpretation must be performed within the context of the patient's risk factors, symptoms, concomitant conditions, medications, and the physical findings. So, whatever interpretation we are giving to a particular patient should be correlated with the patient's history, his risk factors that he has been exposed to, the symptoms he is showing, and the concomitant conditions he is into. So there the different laboratories will give the different amount of normal values for the in the normal range. So there is no need to panic about that also. All right. So no one test enables the clinician to accurately assess the liver's total functional capacity. To increase the sensitivity and specificity, we have to use them as a battery of tests, as a battery cannot deploy a different cells. All right, so it has to function all together to perform its vital function. Same is with the liver, that all of its functions have to be deployed in association with the other functions to perform well. Otherwise it won't be able to give its proper functioning. So when one test provide abnormal finding or persistently abnormal on serial determination, probability of liver disease is high. All right. So if there are abnormal findings of the test that you have performed, then it is probably the abnormality in the serial determination. Serial determination should be made so that to know that what kind of liver disease is there or not. So, when all results are normal, probability of missing occult liver disease is low. So, commonly employed tests in the liver function are bilirubin test, aminotransferase enzyme test, alkaline phosphatase test, albumin, and the prothrombin time. So, let's talk about some of the standard liver function tests that are there. So, as I told you that numerous laboratory investigations have been proposed in the assessment of liver diseases. So, a standard liver function test will help to detect the abnormality and extent of the liver damage. So, the classification is based on the specific functions of the liver that is involved in it. First of all, the liver function test consists of the tests that are based on the metabolism. All right. So the tests that are based on metabolism are first of all, it is based on the carbohydrate metabolism, which includes the glucose tolerance test and the fructose tolerance test. Thereby, the second one is the it is based on the lipid metabolism. So tests that are based on the lipid metabolism are the serum cholesterol, which evaluates the measurement of the free and esterified form of cholesterol estimation and their ratio. And the second one is the estimation of the fecal fats that is there. All right. Third category, which is based on the metabolism, is of the protein metabolism. So in the protein metabolism category, there is estimation of total protein, albumin globulin, and the albumin globulin ratio. In the protein metabolism, there is also determination of prothrombin time and flocculation test that is done for which includes the thymol turbidity test or the zinc sulfase test, colloidal gold test, cephalin cholesterol flocculation test, etc. Amino acids are also assayed in the urine in the metabolism of protein. Next, classification of the liver function test is based on the detoxification and protective function of the liver. So detoxification and protective function of the liver includes tests such as first of all, it includes the conversion of ammonia to urea. So to evaluate the conversion of ammonia to urea, estimation of blood urea in the in blood urea in the laboratory is performed. Secondly, estimation of blood ammonia is performed. Then formation of bilirubin diglucuronate is also assayed. For that, we have to estimate the serum bilirubin in which is there in the body, both directly and indirectly. Icteric index is calculated. Vandenberg reaction is performed to check the presence of bilirubin in the body. Then urinary estimation of bilirubin and urobilinogen is also performed. Next are the tests which are based on the excretory functions of the body. So the tests which are based on the excretory function of the body are bromosulfathaline test, which is also called as the BSP in abbreviation, and second one is the I-131 rose bengal test, which is used to determine the excretion of the rose bengal dye from the Kupffer cells of the liver. Next are the tests which are based on the this storage function of the liver. So store on the basis of storage function of the liver, glycogen estimation is performed, lipid estimation is performed, and estimation of vitamin A, D, and B12 is performed. Next, the serum iron and the serum iron binding capacity can also be performed to test the storage function of the liver, that whether the liver is storing the components that it has been deployed for storing of the by the liver or not. Next are the tests which are based on the serum enzymes which are derived from the liver, which do not directly tells about the functioning of the liver, but it tells about the damage that has occurred to the liver cells. So first of all, transaminases are evaluated. Then the aminotransferases such as ALT and AST are determined. AST is the enzyme which has its source from liver, cardiac muscle, skeletal muscle, kidneys, brain, pancreas, lungs, leukocytes, and the RBCs. Whereas the ALT is has source from liver only. All right. So liver cell damage is evaluated by the aminotransferases enzyme. It has increased permeability and in the increased serum level in case of any kind of damage to the liver. But it has a poor correlation between the liver cell damage and the level of AST and ALT. All right. Next is the cellular structural the next enzymes that are tested for the livers are the alkaline phosphatase, S-nucleotidase, and the gamma glutamyl transpeptidase enzymes. All right. So these are also increased in the case of the damage of the liver. Next, liver function tests that are performed are the cellular structural studies, such as the liver biopsies performed to check the structural organization and the architectural presence of the liver. Next, why these liver function tests are important. So, first of all, to assess the severity of the liver damage, that up to what extent the liver has been damaged by a certain disorder or dysfunctioning that has been there in the body. Next, one is to differentiate between different types of jaundice. We also perform the liver function test to know which kind of jaundice is there, or if the person is suffering from jaundice or not. Next, to rule out the presence of latent latent liver disease if there is any. So the first group of tests includes the test regarding the secretory, excretory, and enzymatic functions of the liver, such as the serum bilirubin test, bilirubin and urobilinogen in urine, the BSP excretion test, serum alkaline phosphatase estimation, and the SGPT. So these are performed to rule out the presence of latent liver disease. The second group of tests that is performed are meant for assessing the protein synthetic functions such as total protein estimation, albumin, globulin, Ag ratio, and the prothrombin time. Then the final group of tests include the tests that are meant for lipid metabolic functions of the liver. So the estimation of serum cholesterol and determination of free and esterified cholesterol ratio is performed in this final group. Next are the tests that are based on the abnormalities of the bile pigment metabolism. So to rule out the any kind of abnormalities in the bile pigment metabolism, different types of tests are performed. First one is the Vandenberg reaction and the serum bilirubin. So bilirubin is usually estimated by the Vandenberg reaction, which involves the diazo reagent. The Vandenberg reaction consists of two parts, the direct and the indirect reactions. So the direct reactions include the immediate development of violet color in 10 to 30 seconds, and delayed direct reaction in which color appears from 5 to 30 minutes and then develops slowly to a maximum. Then there is indirect reaction, which is essentially a method for a quantitative estimation of serum bilirubin. In the indirect reaction, serum is diluted with distilled water, and methanol is also added in a small amount to precipitate the proteins. But it is not sufficient to precipitate the protein, but it is sufficient to permit all the bilirubin to react with the diazo reagent, and thus we get the indirect evaluation of the bilirubin. Bilirubin, as you can talk about it, that it is insoluble in water, while the unconjugated bilirubin is soluble and gives direct positive Vandenberg reaction. The unconjugated bilirubin gives indirect positive Vandenberg reaction. If in case the serum contains both unconjugated and the conjugated bilirubin high concentration, the purple color is immediately produced, which is further intensified by the addition of any kind of alcohol. So this type of reaction is also known as biphasic reaction. So based on it, the normal serum bilirubin ranges between 0.2 to 0.6 milligram per deciliter. So the Vandenberg reaction can be seen in different forms in different types of jaundice. For example, in hemolytic or pre-hepatic jaundice, unconjugated bilirubin is increased. Thus, we can say that indirect positive reaction is obtained. All right. Next, in obstructive kind of or post-hepatic jaundice, conjugated bilirubin is increased, thus an immediate direct positive reaction is obtained. And in hepatic jaundice, either or both may be present, which will involve the biphasic kind of reaction. All right. So tests that measure the biosynthetic function of the liver are also performed. These includes the coagulation factors such as except for factor 8, blood clotting factors are exclusively synthesized in the hepatocytes. The rapid turnover is the measurement of clotting factors is the single best accurate measure of the hepatic synthetic functions of the liver. So the tests that are performed for the biosynthetic functions of the liver includes the measurement of prothrombin time. Why prothrombin time is measured? Because it gives the collective measure of second coagulation factor, fifth coagulation factor, seventh coagulation factor, and the tenth coagulation factor. So the prothrombin time is elevated in case of different conditions such as hepatitis, cirrhosis, and disorders that will result in the deficiency of the vitamin K. If we are observing more than 5 seconds above the control prothrombin, then it is not corrected by vitamin K. All right, so it is a poor prognosis which is seen in any kind of acute viral hepatitis and other acute and chronic liver disease. So hemolytic or prehepatic jaundice gives the isolated elevation of bilirubin. Bilirubin is normal to 5 mg per deciliter, and no bilirubinuria is seen here. Then the fractionate bilirubin is there. The fractionate bilirubin consists of the direct bilirubin more than 15%, and direct bilirubin can be less than 15%. So a direct bilirubin less than 15% will be there in the case of hemolysis. All right. There is the flow chart of hepatocellular hepatic jaundice also. Bilirubin is increased, both fractions, direct and indirect. Bilirubinuria is there. ALT and AST are increased. ALP can be normal and can also be increased up to threefold depending upon the severity of the damage. Then albumin may be normal, albumin may be decreased, PT is normal, and PT can also be prolonged with failure to correct. In obstructive also there is one chart that is being shown to you. So basically there are different tests that can be assessed for the diagnosis of the liver dysfunctioning that are there in the liver. So what is the value of serum? Let's discuss value of serum enzymes in the liver diseases. So a large number of enzyme estimations are available which are used to ascertain different liver function tests. Most routinely used laboratory tests are the two, which is serum transaminase, which is also called as the aminotransferase, and the serum alkaline phosphatase. So the normal level of serum alkaline phosphatase is between 3 to 13 king-angstrom units per 100 ml. All right. So the increased level of alkaline phosphatase is found in post-necrotic disease, cirrhosis, carcinoma of the liver, obstructive jaundice, hepatocellular jaundice, etc. So, why we perform liver biopsies? So, liver biopsies are performed in the case of any kind of fibrosis or neoplasm of the liver. So, histopathological studies of the liver biopsy reveal various pathological states of liver cells, and it also gives the indication for the liver biopsy as fibrosis or neoplasm of the liver is seen in metabolic diseases, arsenic poisoning, selenium poisoning, and for estimation of vitamin A, E, copper, zinc, liver, glycogen. The liver biopsy is taken and it is being performed. So biopsy is also contraindicated in case of any kind of liver abscess. So let's sum up it. So ALT, what it tells us about the hepatocellular damage. AST also tells us about the hepatocellular damage. The bilirubin assay gives us the fair idea about the cholestasis and the impaired conjugation or the biliary obstruction of the liver. Then the ALP is there to give us an idea about the synthetic function that is performed by the liver, if the function is performing its synthetic function well or not. Next is the albumin. It is also measured for knowing the synthetic function of the body. Gamma glutamyl transpeptidase is there to indicate any kind of disease or suspect any kind of disease such as cholestasis or the biliary obstruction, which includes the adhesion of the fat layer to the liver. Then the assay of 5-nucleotidase also gives an idea about the cholestasis or the biliary uh, obstruction of the liver. So that's all about the liver function test, students. If you have any question or any queries related to today's session, you may ask in the chat box. The chat box is available to you. I'm there to answer all your questions. Students, if you have any query, you may write down your questions in the chat box. So yeah, that's all for today's session, students. If you have any query, you may still write to the feedback section that is there on your LMS. Thank you for joining with us. Now you may end this session. Hello students and welcome to the study portal. The topic of today's online class is cardiac function test. So in today's online session we are going to study about the functioning of the heart, that how the heart performs its function well and how we can evaluate this functioning of the heart, that the heart is properly performing its functions or not. So without delaying, let's start with today's session. So first of all, let's have an introduction with the cardiac function test or the cardiac profile test. So, if we talk about the cardiac profile test, it is a comprehensive panel of tests which is used to check the overall functioning of the heart. By looking at the concentration of fats, certain enzymes, and the composition of urine and blood, it becomes easier for the clinicians to evaluate the functioning of the heart. So we can say that cardiac function tests are the panel of tests, are the it is like a proper array of the tests which are used to determine whether there is any cardiac tissue damage or the damage to the heart or not, whether your heart is performing its all of its functions properly or not. Right, so it measures the levels of the cardiac biomarkers also in the blood, which includes the enzymes, hormones, and the proteins. They show up in the blood after the heart has been under severe stress and becomes injured because it is not getting enough oxygen, etc. Like, right, this so it is performed to help diagnose different types of cardiac diseases such as myocardial infarction, coronary artery disease, and atherosclerosis. Right, so what all are included in the cardiac function test? Let's have an overview. The cardiac function test or the cardiac profile includes the tests such as CK, that is creatine kinase enzyme, then the CKMB, which is creatine kinase muscle brain, then LDH, which is lactase dehydrogenase, SGOT, serum glutamate oxalotransaminase, troponin test, then the myoglobin test. Lipid profiles also comes under the cardiac function test, which includes the evaluation of cholesterol, triglycerides, HDL cholesterol, LDL cholesterol, and the VLDL cholesterol. Thereafter, LDL-HDL ratio, it also comes under the lipid profile. LDL total cholesterol ratio, it also comes under the lipid profile. Then apolipoprotein, which is A1, and apolipoprotein A2. Right, so these are the tests which comes under the cardiac function test. We are going to study all of these tests in detail in today's online session. Right, before that we should know how it is done, how the cardiac profile test is performed. So it is basically enzyme marker test or the lipid profile test, so which requires a blood sample. The blood is drawn from the patient, right, and then it is tested in the laboratory. What do cardiac blood tests show? These cardiac blood tests show, or these are performed to check if there is anything in the blood that might indicate a heart attack or other cardiac illness. It also measures the level of substances such as cardiac markers that are present in the blood, or such as blood fats and minerals, to work out any future risk of any kind of heart disease. Right, this is clear that what all is cardiac profile test and what all tests are included in the cardiac profile test. So without delaying, let's begin with the cardiac functioning test. Right, so the first enzyme which is tested is CK enzyme. CK enzyme is the creatine kinase enzyme. Here you can see the classification, principle of this CK creatine kinase enzyme test. So let's start. So creatine kinase, which is also known as creatine phosphokinase or phosphocreatine kinase, it is an enzyme which is expressed by various tissues and cell types. Right, CK catalyzes the conversion of creatine and uses adenosine triphosphate to create phosphocreatine and adenosine diphosphate. This CK enzyme is reversible and thus ATP can be generated from it. Right, so in tissues and cells which consume ATP rapidly, especially skeletal muscles, but also brain, photoreceptor cells of the retina, hair cells of the inner ear, etc., they serve as an energy reservoir for the rapid buffering and regeneration of ATP. Right, so creatine kinase is an important enzyme in such tissues. Right, So here let's look at the principle that how this test is performing its mechanism. So creatine phosphate uses ADP in the presence of CK enzyme, and then it converts into creatine plus adenosine triphosphate. Then the D-glucose and ATP in the presence of hexokinase enzyme gets converted into D-glucose 6-phosphate and adenosine diphosphate. The D-glucose 6-phosphate and NADP gets converted into D-gluconate 6-phosphate and NADPH plus H in the presence of G6PD-H enzyme. Right. So the method of testing is increasing kinetic. The normal range of CK enzyme is 25 to 120 units per liter. Now let's talk about the clinical significance of this enzyme. So what is the clinical significance? That the CPK enzyme activity is increased in the cases such as cerebrovascular stroke, myocardial infection, acute coronary syndrome, any kind of crush injury, myopathy, and polymyositis. So if the biological reference range is more than 120 units per liter, then it is suggestive of diseases like cerebrovascular stroke, myocardial infection, acute coronary syndrome, etc. Right. Now talking about the second test, which comes under the cardiac profile test. So the second test which comes under the this cardiac profile test is CKMB, which is also known as CKMB test. It is a cardiac marker which is used to assess the diagnosis of an acute myocardial infarction, myocardial ischemia, or myocarditis. It measures the blood level of CKMB, the bound combination of two variants of the enzyme phosphocreatine kinase. Right. There are many cases of CKMB levels exceeding the blood levels of total CK have been reported, especially in newborns with cardiac malformation, etc. Right. So it is one of the cardiac marker test. Right. This is clear to everyone. So if we talk about CKMBs, it is a subtype of CK creatine kinase enzyme. It is more sensitive for finding heart damages from a heart attack. It rises 4 to 6 hours after a heart attack, but it is generally back to normal in a day or two. Because of this, it is not helpful when a healthcare provider is trying to figure out if your recent chest pain was a heart attack. Right. So this antibody completely inhibits the activity of CKMM and half of the activity of CKMB, while not affecting the subunit B activity of CKMB and CKBB. What is the method of evaluating the CKMB? The method is increasing kinetic UV method. Right. The normal range for this is 0 to 25 international units per liter. What is the significance? That it rises, it is present only in cardiac tissues, so it is specific for the diagnosis of cardiac disease, and it rises in the 4 to 6 hours after a heart attack. Right. This is clear. So creatine kinase dimer, it is of the three isotypes: CK1, which is CKBB. The composition of its brain, and the location is brain. Then the CK2 isoenzyme, which is also called as CKMB, which is present in muscle and brain. The location is myocardium. Then CK3, that is CKMM, which is muscle, and it is located in the skeletal muscle. Right. Now talking about LDH, which is also called as lactose dehydrogenase enzyme. Lactose dehydrogenase enzyme also comes under the cardiac marker test, right? So it catalyzes the conversion of pyruvate to the lactate. It is an important enzyme which helps with the cellular respiration, the process through which the body transforms the glucose from the food you eat into energy for your cells. These enzymes are proteins which help to speed up the metabolism or the chemical reactions in the body. Right, this is clear. So what does it do? It catalyzes the conversion of lactate to pyruvate and then back, as it converts the NAD plus to NADH and back. So it is an enzyme which transfers a hydride from one molecule to the another. It also exists in 4 distinct enzyme class. Right. So this is how the LDH is performed. So the method to perform the LDH is IFCC method, which is lactate to pyruvate conversion method, and the increasing kinetic method. If we talk about the normal range of LDH, then the normal range of SDH is between 70 to 240 international unit per liter. Right. What is the clinical significance? If the level of LDH rises from this, then it may be suggestive of prognostic tumor marker, or if in case of RBCs it may be suggestive of malaria, sickle cell anemia. In case of liver it may be suggestive of viral hepatitis. In case of cardiac tissue it may be indicative of myocardial infarction, right? So these are the five isoenzymes of the LDH. The five isoenzymes of LDH are LDH1, LDH2, LDH3, LDH4 and LDH5. The location are given. So the LDH1 is 20 to 30% of total, and it is located in the myocardium. The LDH2 is 30 to 40% of the fraction of total LDH, and it is located in the RBCs. LDH3 is 20 to 25% of the total fraction, and it is located in the lung. LDH4 is 10 to 15% of the total LDH, and it is located in the kidney and pancreas. LDH5 is 5 to 15% of the total fraction, and it is located in the skeletal muscle and the liver. Now talking about the enzyme SGOT or AST in the cardiac marker test, right? So SGOT is also called as the serum glutamate oxalotransaminase enzyme. The levels of SGOT are very important for diagnosing the any kind of myocardial infarction, or to detect this enzyme. So it is important to note that the cardiac enzymes such as creatine kinase, AST and LDH are of variable specificity to the cardiac muscles, right? So cardiac, what does it do? The principle is of this test is that the L-aspartate and alpha ketoglutarate, when they act or get mixed in the presence of SGOT enzyme, serum glutamate oxalotransaminase enzyme, then it gets converted into oxaloacetate and L-glutamate. Then the oxaloacetates in the gets reacted with the NADH in the presence of MDH enzyme and converts it into malate. The malate dehydrogenase converts the oxaloacetates into malate and NAD+, right? So the rate of NADH to NAD is measured as a decrease in absorbance at the 340 nanometer. The method to perform this test is like IFCC method, with or without the pyridoxal 5-phosphate, and the decreased UV method also. Right, the normal range of SGOT is 15 to 45 international unit per liter. Right, so after any kind of congestive heart failure, there is a rise in the SGOT level due to less blood supply to the liver. Right, there is a rise after 6 to 8 hours of the damage or chest pain in the myocardial infarction also. Right, so this is the clinical significance of the SGOT enzyme, because it is like it has a significant importance in case of acute hepatocellular damage, myocardial infarction, congestive heart failure, biliary tract obstruction and the cholecystitis. Right, so aspartate AST, why it is called as AST? Because of the aspartate transaminase, right? This is where it plays a role. It plays a role in the liver cell injury. It plays a role in the biochemical reaction in the myocardial infarction, ischemia, etc. Right. Now talking about other enzyme, which is called as the cardiac troponin 1 and the cardiac troponin 2. Cardiac troponin are the biomarkers, the cardiac biomarkers which are present in the body that are evaluated for checking the risk of any kind of heart disease. So troponin is a type of protein which is found in the muscles of the heart. It is not normally found in the blood, but when heart muscles become damaged, troponin is sent into the bloodstream. As heart damage increases, the amounts of troponins are increased, increased amount of troponins are released in the blood. right? So, cardiac troponin is a biomarker of choice for the diagnosis of myocardial necrosis because it is the most sensitive and specific biochemical marker of myocardial injury or the necrosis that is available. Right? Troponin or troponin complex is a complex of three regulatory proteins which are integral to the muscle contraction in skeletal muscle and cardiac muscle, but not the smooth muscle. So the measurement of cardiac specific troponins I and T are extensively used as diagnostic and prognostic indicators in the management of myocardial infarction and acute coronary syndrome. So blood troponin levels may be used as a diagnostic marker for stroke or other myocardial injury which is ongoing, although the sensitivity of this measurement is low. So, the troponin is attached to the protein tropomyosin and lies within the groove between actin filaments in the muscle tissue. If we talk about the relaxed muscles, the tropomyosin blocks the attachment site for the myosin crossbridge and thus it prevents the contraction. So when the muscle cell is stimulated to contract by an action potential, the calcium channels open in the sarcoplasmic membrane and releases the calcium into the sarcoplasm. Some of this calcium gets attached to the troponin which causes it to change its shape, exposing the binding sites for myosin. The myosin's binding to actin causes cross-bridge formation and contraction of the muscle begins. So, troponin is found in both skeletal muscle and cardiac muscle, but the specific version of troponin differs between the types of muscles. The main difference is that the TNC subunit of troponin in skeletal muscle has 4 calcium ion binding sites, whereas if we talk about the cardiac muscle, there are only 3, right? So this is why the cardiac troponin 1 and cardiac I and cardiac troponin T are important biomarkers for the cardiac marker for the cardiac diseases or disorders that are there. Right, so this is how they work. The troponin I binds to the actin in thin filaments to hold the actin tropomyosin complex in place, whereas if we talk about the troponin T, it binds to tropomyosin, interlocking them to form a troponin tropomyosin complex. Right, so the cardiac troponin I and T are measured by ELISA or RIA techniques, immunoturbidometry test, and the immunodiffusion method. The serum level of troponin T increases within 6 hours of myocardial infarction, but troponin I is released into the blood within 4 hours after the onset of cardiac symptoms. Right. Normal range of troponin I is between 0.04 to 14 nanogram per milliliter, and troponin T is 0.01 to 25 nanogram per ml, increasing which it may be suggestive of any kind of myocardial damage. It is most sensitive in specific tests. We can early detect the presence of heart disease mainly in the case of chest pain, severe acute chest pain. It is usually mainly in the diagnosis of chest pain when patient's ECG is normal. Right, so this is the graph which is showing the enzyme activity after the myocardial infarction. So the time is given at the x-axis and at the y-axis enzyme activity is given. So here you can see in the first hour troponin levels are high, the CK-MB level are high, I had specific LD and the total CK. Then in second hour the troponin levels are decreased, the CK-MB level are decreased, LDH level are increased, but the CK levels can be seen as elevated. So with the increase, if we talk about the early diagnosis, for the early diagnosis, troponin is the most sensitive test because the enzyme activity, the troponin activity is highest in the initial hours. Right, so these are the plasma enzyme changes after myocardial infection. There is given the enzymes and the abnormal activity in the terms of R. So C troponin, cardiac troponin 1 I is abnormal activity is shown within 4 to 6 hours. The peak value R is 12 to 24 and the duration remains for 10 to 14 days. Next is CKMB. So CKMB is also detected with between 4 to 6 hours of the peak value is 12 to 24 and the duration remains for 1.5 to 3 days. Total CK, it is like diagnosed after 6 to 12 hours and the peak value is 18 to 30 hours and it remains for 2 to 5 days. AST is also detected for 6 to 12 hours, right? And the peak value remains for 20 to 30 days and it remains for 2 to 6 days. LDH1, the abnormal activities is diagnosed between 8 to 18 hours. The peak value is 30 to 48 hours and it remains in blood for 5 to 14 days. Now talking about the myoglobin, which also comes under the cardiac profile test. So what is myoglobin? First of all, understanding this is important. So myoglobin is a protein which is found in the striated muscles, which includes the skeletal muscles and the heart muscles. Its main function is to supply oxygen to the cells in the muscles, which are called as the myocytes. So all cells in the body needs oxygen in order to function. So to transport the oxygen to the muscle cells, myoglobin protein is there, right? It is the oxygen binding protein, right? It is released mainly from the skeletal muscle damaged tissue. Its level rises more rapidly than the cardiac troponin and the CKMB, right? What is the role? You see that the myoglobin facilitates oxygen diffusion. It desaturates at the onset of muscle activity, which increases oxygen's diffusion gradient from the capillaries to the cytoplasm, right? It has also been shown to have enzymatic function, right? So why it is important in the cardiac test? Because its levels rises more rapidly than the cardiac troponin test. It is evaluated by RIA, radioimmunoassay, ELISA and the chemiluminescence method. The normal range of myoglobin is between 25 to 72 nanogram per liter, and if the levels rises more than this, then it is suggestive of major skeletal muscle injury, crush injury or any kind of burns. Now talking about the lipid profile. So lipid profile in lipid profile test is like solo videos is also available where you can see. So the lipid profile test contains a number of tests, a parameter of tests such as total cholesterol, triglyceride, HDL cholesterol, LDL cholesterol, VLDL cholesterol and the ratio of the LDL and VLDL also. So these are the lipid profile tests which also comes under the cardiac profile test, because if the cholesterol level or triglyceride levels are high, then it is suggestive of any kind of cardiac damage or any kind of cardiac disorder, right? So this is how these are performed. The increased cholesterol level are suggestive of increased probability of atherosclerosis, coronary artery disease, ischemic heart disease, cerebrovascular stroke, hypertension and the xanthoma. Whereas if the cholesterol is decreased than its normal range, then it may be suggestive of depression. The normal range of cholesterol is less than 199 mg per deciliter in normal, and in borderline it is 200 to 239 mg per deciliter. Now talking about triglyceride. So increased triglycerides are suggestive of borderline hypertriglyceridemia or increased risk of pancreatitis as well. The normal range of triglycerides in blood is less than 150 mg per deciliter. So this is how the HDL, LDL, VLDL are related with liver cell and the liver and the cell. So what happens? The liver is there which converts the HDL into bile salts. Then the HDL is after conversion is transported to first of all let's talk about it. So cell VLDL gets transformed into LDL. Then the LDL delivers the cholesterol to the cells. From here the HDL removes the excess cholesterol from the cells, and then this HDL in the liver gets converted into bile salts, right? So HDL is a high density lipoprotein, right? It is detected by precipitation reaction. It precipitates all lipoprotein other than HDL, right? So increased amount of or any abnormal amount of HDL, it is it transports the cholesterol from peripheral tissues to the liver. It also helps in the removal of macrophages from the arterial walls. The abnormal levels of HDLs are indicative of high risk of heart disease also, right? If the HDL levels are less than 40 mg per deciliter, it means that there is a high risk of heart disease. Then low density lipoprotein is there, right? Low density lipoprotein, it means if the levels are higher than 160 mg per dl, then it is suggestive of heart disease, right? Now talking about VLDL is very low density lipoprotein, right? If the amount is 160 and higher than that, then it may be suggestive of heart disease, right? So now talking about the apolipoprotein A1 and B. So it is it is more than 50 mg per dl, then it is desirable. But if it is less, then it is suggestive of heart disease or heart disorders. So that's all about today's session. Hope student you have liked today's session. Keep watching the online session videos that are there. Hello students and welcome to the study portal. The topic of today's online session is renal function tests. So what are renal function tests? So renal function tests are the tests that measure the ability of the functioning of your kidneys. So in today's session we'll be discussing about the kidneys, that what are kidneys. We will be having an introduction about the kidneys. Thereby we'll be talking about the different functions that are performed by the kidney, and then we will discuss about the renal function tests that are performed in the laboratories or hospital for the diagnosis of the functioning of the kidney. We will study about the different parameters that are there in the renal function test and what is the clinical significance of those parameters that are included in the renal function test. So without delaying, let's start today's session. First of all, what are kidneys? So kidneys are the organ, as we all know, which filter waste products from the blood. It is a important organ of the body, which has its primary function in the removal of the waste products from the blood. So there are one pair of kidney. There are two kidneys which are reddish brown or bean shaped organ, which are found in the humans. They are located on the left and right in the retroperitoneal space, and in adult humans they are about 12 centimeters in length. All right. They receive blood from the paired renal arteries. Blood exits into the paired renal vein from the kidney. So this is clear that the kidneys are a pair of bean shaped organs on either side of the spine, below your ribs, all right, and behind the belly. Each kidney is about 4 or 5 inches long, roughly the size of a large fist, we can say. So the kidney perform three essential functions. First one is the removal of the metabolic waste products and toxins. Second is the regulation of the body fluids status, electrolyte balance, and the acid-base balance. And the third one is the production or activation of hormones that are involved in the process of erythropoiesis. All right, erythropoiesis, that is the formation of red blood cells, the calcium metabolism, and the regulation of blood pressure and blood flow. So first of all, the primary function of the kidney is to filter the blood. It removes the waste, control the body's fluid balance, and keep the right levels of the electrolytes. All right, so all of the blood passes through the kidney about 40 times a day. It comes to the kidney, waste gets removed, and salt, water, and minerals are adjusted if it is needed. Then the filtered blood goes back into the body. The waste get turned into urine, which collects in the kidney's pelvis, which is a funnel-shaped structure, which drains down a tube called the ureter to the bladder. So each kidney has around a million tiny filters, which are called nephrons. You could have only 10% of your kidneys working, and you may not notice any symptoms or problems. All right. This is clear that what is kidney and what is its function. Its function may be assessed by measuring the components that are there in the urine such as or blood such as urea and serum creatinine. So renal function decreases with age, which must be taken into account when interpreting the test results. Alright, so what are renal function tests? Now there comes the question that what are renal function tests? So renal function tests are urine or blood tests which evaluate how well the kidneys are performing. Alright, so these are the group of tests we can say which are performed to evaluate the functioning of the kidney, that if the kidneys are performing their functions well or not. So these tests measure levels of various substances including several minerals, electrolytes, proteins and glucose in the blood as well as in the urine to determine the current health of the kidneys. So if the kidneys are not functioning properly, waste products can accumulate in the blood and fluid levels can increase to dangerous volumes. So which will cause a damage to the body or a potentially life-threatening situation. So numerous conditions and diseases can result in the damage to the kidney. Alright, so the most common causes of and main risk factors for kidney disease are the diabetes and hypertension. We all know this. Right? So RFTs gives us information regarding the renal blood flow, if the blood flow to the kidneys is proper or not. The GFR, which is the glomerular filtration rate, we'll be discussing about it. The renal glomeruli function, that the glomerulus that is there in the kidney is performing its function well or not. The renal tubular function, that the tubules in the kidneys are functioning well, they are filtering the blood properly or not. The urinary output, if the urinary output is proper in a particular person or not. So the tests which are involved in the RFT renal function test are urea, ammonia, parathyroid hormone, calcium, uric acid, potassium, creatinine clearance and the glomerular filtration rate. We will be discussing about all of these components in detail in today's session. So first of all, before moving on to these tests, what are the indication for the assessment of these kidney function tests? That why these kidney function tests are performed? What are the indications in which a doctor may prescribe a renal function test? So the indication for the assessment of renal functions are varied and range from acute emergency to the chronic settings. First of all, primarily renal function tests are performed to identify the renal diseases, to determine appropriate patient management and prevent further deterioration of the renal function. Further indications in patients in whom the renal disease has been identified are to stage the level or type of the renal diseases and to monitor the progression of the renal diseases to ensure that optimal management occurs and to monitor the response to the intervention. The renal function tests are performed. Clinically, these are the most practical tests which are there to assess the renal function test to get the estimation of the functioning of the kidneys. Alright, this is clear. So moving on, now talk about the tests that are involved in the renal function test. One by one. First parameter is the creatinine. So creatinine, what is creatinine? It is a waste product which comes from the normal wear and tear on muscles of the body. in the muscles, the creatine is converted to creatine phosphate, which becomes the source of high energy phosphate bond for the immediate reformation of the ATPs. We can also say that creatinine is the byproduct of the muscle energy metabolism, because it is produced by the wear and tear of the muscles of the body. All right, so it is produced at a constant rate according to the muscle mass of the individual. All right, so creatinine is observed in the renal function test, because creatinine levels in the blood can vary depending on the age and body size. So thus the creatinine is performed renal function test. So it is a substance which is easily excreted by the kidney, because the kidney is excreting the creatinine. So by understanding the levels of the creatinine, we can come to the conclusion that if the creatinine is excreted by the kidney in a normal amount or the abnormal amount, which may indicate the abnormality in the functioning of the kidney. So creatinine is performed by the Jeff's method. So what is Jeff's method? So this is a colorimetric method in which the creatinine in the serum is reacted with the picrate ion, which are formed in the alkaline medium to develop a red-orange color. The color which is produced by during this reaction is then compared in a colorimeter, which is an instrument which measures the color concentration of the solutions. All right, by understanding the color concentration of the solution, and the measurement of the creatinine is performed. Thereby, there is a test. There are clearance tests that are used for the kidney function test. The clearance test determines the measurement of the any substance which is eliminated from the blood by the kidney. All right. So creatinine clearance test is one such test which comes under the clearance test. So as we know that creatine is a waste product which is excreted from the kidney. So creatinine clearance test compares the creatinine in a 24-hour sample of a urine to the creatinine level in the blood, which will show how much waste products the kidney are filtering out in each minute. All right, so what is a creatinine clearance test? It is the comparison of creatinine that is excreted by the kidney in a 24-hour sample of the urine with the creatinine levels in the blood, in the serum. What is the creatinine level? So what it will give us, or it will show us that how much waste products the kidneys are filtering out each minute. So the value of the creatinine clearance is given in units of millions per minute, which represents the volume of blood cleared by the kidney per minute. It has a calculation formula also. Creatinine clearance is equal to U into V by P, where U is the urine creatinine concentration, V is the volume of urine in milliliter per minute that is excreted, and P is the serum creatinine concentration in mg per deciliter. Alright, so based on the rate of excretion by the kidneys of metabolically produced creatinine, the creatinine clearance tests are defined. Alright, so it is freely filtered by the glomerulus and is not reabsorbed by the tubules. The reference range or the normal range for the creatinine is, in males it is 0.7 to 1.3 mg per deciliter, while in female it is 0.4 to 1.1 mg per deciliter. If the concentration of the creatinine rises or decreases above or below this reference range respectively, then there may be a suspicion of certain diseases. Alright, so what are the suspected disease that are likely to be present in a person if the creatinine levels are high? So if the creatinine levels are high in the particular person in its blood or like urine, then it may be suspected of having acute and chronic kidney diseases. the obstruction in the ureter, dehydration, and the glomerulonephritis, which is the inflammation of the nephrons. All right. Now talking about the parathyroid hormone. So what is parathyroid hormone and why it is detected in the kidney function test? Let's take a look. So the PTH or the parathyroid hormone, or it is also called as the parathormone, is produced by the parathyroid glands in the body. The parathyroid glands are 4 pea-sized glands in the neck. The parathyroid hormone controls the level of calcium in the blood. Calcium, we all know, is a mineral which keeps our bones and teeth healthy and strong. It is also essential for the proper functioning of the nerves, muscles, and heart. If the calcium blood levels are too low, then the parathyroid glands will release parathormone in the blood, which causes calcium blood levels to rise. If the calcium blood levels are too high, then these glands, the parathyroid glands, will stop making the parathyroid hormones. So the PTH levels that are too high or too low can cause serious health problems, which may include the hyperparathyroidism, hypoparathyroidism, any kind of kidney disease, right? Any kind of kidney stones may also be present in the blood. So why parathyroid hormone is there in the renal function test? Because it assesses the functioning of the kidney, that if the kidney is performing its function well or not. Because in the case of increase or decrease in the parathyroid hormone, suspicion of kidney disease is there. All right. So let's have a look at that. How does the parathyroid hormone regulate the calcium levels in the blood? So if the calcium level, as I told you, that if the calcium blood levels are too low, then the parathyroid glands will they do? They will release the parathyroid hormone into the blood, which will cause the calcium level to rise. The calcium level will rise. All right. So this causes calcium levels to rise. If calcium blood levels are too high, then the parathyroid glands will stop making the parathyroid hormones. So parathyroid levels are too high or too low, it is indicative of any kind of serious diseases. So if increased calcitriol formation decreases excretion of calcium and increases the excretion of parathyroid hormone. Now let's have a look at this role of PTH. The role of parathyroid hormones is to regulate the calcium levels. Its major function is to regulate the calcium levels in the body within a very narrow range of which is 8.5 to 10.2 mg per deciliter. This is the normal reference range for the calcium levels in the blood. All right. Then activation of vitamin D is also essential for calcium absorption from the GI tract, which is also regulated by the parathyroid hormone. So vitamin D has to be get converted into 1-dihydroxy cholecalciferol in the liver and the kidney in the presence of PTH, which is parathyroid hormone. So parathyroid hormone, we can say that it also increases the formation of dihydroxy cholecalciferol. Right. Now talking about uric acid. So what is uric acid? Uric acid is a naturally occurring waste product which resulting from the breakdown of purine crystalline compound, which is found in certain foods. All right. So uric acid is also tested under the renal function test. So it is a heterocyclic compound of carbon, nitrogen, oxygen, and hydrogen. It forms ions and salts known as urates and acid urates, such as ammonium acid urate. So what is uric acid? Uric acid is a byproduct of the metabolic breakdown of purine nucleotides, and it is also a normal component of the urine. Thus, it is also tested under kidney function test. So if there are normal conditions, then the uric acid will dissolve into the blood and will pass through the kidney and is eliminated with the help of urine. But in an abnormal condition, it may not be able to dissolve in the blood and then pass through the kidney, and then it may not be eliminated in the urine. So sometimes the body produces too much uric acid and does not filter out enough of it, and at that time uric acid levels are increased in the blood, so which will give us the functioning abnormality of the kidney function test. Mainly two types of conditions are observed when the uric acids are below the normal range or above the normal range. So if the uric acid levels are increased in the blood, then the condition is called as hyperuricemia, and if it is decreased in the blood, then the condition is called as hypouricemia. So uric acid blood test is performed, which is also known as the serum uric acid measurement. It determines that how much uric acid is present in the blood. This gives us the determination of the how well our body is producing and removing the uric acid. So it is performed in the laboratories generally. What is the clinical significance? That the uric acid is a byproduct of the metabolism of purines, nucleic acids, and the nucleoprotein. So consequently, if there are abnormal levels of the uric acid, then it may be an indication of the disorder in the metabolism or in certain genetic diseases of the person. So what are the causes of the increase and decrease level? So the high levels of uric acid may be in the blood, may be indicative of diseases like diabetes, gout, which is also called as the acute arthritis, chemotherapy and radiation, leukemia, which is the cancer of the WBCs, hypoparathyroidism, the kidney stones, multiple myeloma, and the metastasized cancer. Whereas the low levels of uric acid in the blood may be found in the diseases such as Wilson's disease, Fanconi syndrome, alcoholism, liver or kidney disease, and a low diet in purines. So it is like assayed in the laboratories by chemical method and the enzymatic method both. The reference range for this serum uric acid in for women it is 2.5 to 6.5 mg per deciliter, and for men it is 3.6 to 7.7 mg per deciliter. Now talking about the urea, which is the chief nitrogenous waste of the body, which is excreted out in the urine. It is the normal component of the urine. It is a naturally occurring molecule which is produced by the metabolism of protein, and it is abundantly found in the urine of the human beings. All right. So urea is the end product of the protein metabolism, and after filtration by the glomeruli, it is partially absorbed by the renal tubules. There are two methods to perform urea estimation in the blood. First one is the kinetic method and second one is the enzymatic method. So generally urea is estimated by the colorimetric method. For colorimetric method, this is the calculation, that how calculation can be done. And the serum if we talk about the reference range of the urea in the blood, then the normal reference ranges of urea is 13 to 45 mg per deciliter in the blood. Urea clearance test is also performed, which will tell us that how much urea is excreted in the urea concentration of the urine that is cleared of urine and blood that cleared by the kidney or excreted by the kidney is compared, and which will give us the complete clearance of the urea per minute by the kidney. So urea clearance test is less than the glomerular filtration rate, and it is highly impacted or influenced by the protein content that is intaken by the person in the diet. So approximately 40% of filtered urea is normally reabsorbed by the tubules, while the rest is excreted out. All right. So sensitivity is much less than the creatinine clearance because plasma concentration of urea is affected by a number of factors like dietary protein, fluid intake, infection, surgery, etc. So the normal value of urea clearance is 75% milliliter per minute. So we can define this as the volume of plasma that would be completely cleared of urea per minute. So it is calculated by this formula which is there for the creatinine clearance also, U into V by P, where U is the urea concentration in urine, V is the urine which is excreted per minute in the milliliters, and P is the urea concentration in the plasma. If the output of urine is more than 2 milliliter per minute, then it is referred to as the maximum urea clearance. Alright, so if the urea clearance level or value is below 75% of normal, then it is very serious. It is indicative of renal damage. Alright, so blood urea level is also found to increase only when the clearance falls below 50% normal. Normal level of blood urea is 20 to 40 mg per deciliter. Now, what are the causes for increased blood urea? So, there may be prerenal condition, renal condition that may in which the blood urea may be increased. So, the reason may be dehydration, severe in case of severe vomiting, diarrhea, diabetic coma, severe burns, severe infection, that dehydration may be there. So, in case of dehydration, also the urea concentration may rise above its normal range. In if renal conditions are there, so in renal condition, in case of acute glomerulonephritis, nephrosis, malignant hypertension, and chronic pyelonephritis, the blood urea level may be increased in the body. Now, what are the conditions that in which the blood urea may be decreased? So, it also may be decreased in late pregnancy, in case of starvation, also in patients which are grossly deficient in protein and which who may have the hepatic failure. So, what is azotemia? This is the word. So, increase in the blood level of NPN, which is creatinine, urea, and uric acid, is referred to as azotemia, and it is the hallmark of the kidney failure. Hallmark of the kidney failure means if azotemia is there, then like 90% there are chances that the person is suffering from the kidney damage up to severe extent. So NPN is non-protein nitrogenous substances such as creatinine, urea, and uric acid. Now talking about calcium, so we all know calcium is a mineral which plays an important role in the transmission of the nerve impulse, muscle contraction, the growth of the bones and teeth, pancreatic insulin release. It is also a core factor for some enzyme reactions and blood coagulation. Alright, so normal total calcium value in the blood is between 8.8 to 10.3 mg per deciliter. So if the calcium has reduced from the normal range, then it is usually an implication of the deficiency in either the production of the calcium or response to the parathyroid hormones. Hypercalcemia is a condition in which the calcium is increased in the blood, and it is usually associated with the malignancy or the metastatic diseases. So these are the reference ranges. You can see from here that what are the reference ranges of calcium in the blood in different ages also. Now talking about potassium. So potassium is also a mineral which is most abundant intercellular cation. Alright, so approximately 3500 milliequivalent of potassium is there in the average 70 kg adult. Only 10% of the body potassium is extracellular. Alright, so what is the role of the potassium? The potassium plays an important role in proper electrical conduction in cardiac and skeletal muscle, and also in maintaining the electrolyte balance in the body. So the potassium is also used to monitor the electrolyte imbalance in the body and the treatment of disease condition which may be characterized by low or high level of the potassium. So it is regulated by the kidneys, aldosterone, arterial pH, insulin, potassium intake, sodium delivery to the distal tubules. Thus, as it is regulated by the kidney, so the low or high level of the potassium may indicate the abnormality in the functioning of the kidney, that the functioning of kidney is impaired, and the kidney is not properly regulating the potassium. So potassium for assaying the potassium, the ion selective electrodes are used, which will develop an electrical potential, which is then measured, and the potassium is measured in the serum. So in the serum, the potassium level is 3.3 to 5.5 millimole per liter. In plasma, it is 3.3 to 4.5 millimole per liter, and urine it is 25 to 125 millimole per liter, depending upon the diet that has been intaken by the person. So, hypokalemia, which is the decreased level of potassium in the blood, these may be there. The conditions can be the kidneys, the improper functioning of the kidney. So, because the kidney is responsible for approximately 90% of the daily potassium loss. So, if the reduced potassium concentration is observed in the blood, then it may be because of the improper functioning of the kidney. In certain cases such as vomiting, diarrhea, or adrenal gland disorders, the hypokalemia may also be caused. Now talking about ammonia. So ammonia is also an important parameter which is estimated in the renal function test. All right. So it is also measured into the in the blood to diagnose the renal functionality. So the ammonia produced in the kidney is either excreted into the urine or returned to the systemic circulation through the renal veins. So ammonia is excreted in the urine, which promotes acid excretion. It is also returned to the systemic circulation, where it is metabolized in the liver in a HCO3 consuming process, resulting in no net benefit to the acid base homeostasis. Thus, ammonia is also measured in the renal function test. Now, talking about the glomerular filtration rate. So, the glomerular filtration rate is a test which is used to check that how well the kidneys are functioning or working. Specifically, it estimates how much blood passes through the glomeruli each minute. What are glomeruli? So, glomeruli are the tiny filters in the kidneys that filters the waste from the blood. So we can say that glomerular filtration rate is the volume of blood which is filtered from the kidney glomerular capillaries into the Bowman's capsule per unit time. Alright. The GFR is performed in the renal function test or the kidney function test. Alright, so it is the overall index of the kidney function test. This is the most significant test of the renal function test. So normal GFR varies according to the sex, age, and body size, and it also declines with the age. Alright, so the central of the physiological maintenance of the GFR. What is the principle? GFR is equal to the clearance rate when any solution is freely filtered and is neither absorbed nor secreted by the kidney. But GFR is typically recorded in units of volume per time. So millimeters per minutes compared to the filtration fraction. So the rate at which plasma is filtered by the kidney glomeruli gives us the rate of the glomerular filtration rate. Alright, so the normal range of GFR is usually 125 milliliter plasma per minute or 180 liters per day. Alright, so it is like in average 70 kg young adult man, about 3 liters the kidney filter the plasma some 60 times in a day. So what are the factors that will affect the GFR? First of all, it is a change in renal blood flow may affect. Glomerular capillary hydrostatic pressure may affect the glomerular filtration rate. The change in the capsular hydrostatic pressure or osmotic pressure, glomerular capillary permeability, and the size, shape, and electrical changes of the macromolecules may also affect the levels of glomerular filtration rate. All right. Now talk about clearance. What is clearance of this glomerular filtration rate? So the measurement or calculation of the glomerular filtration rate is considered the most sensitive chemical test for assessing the functioning of the kidney. Usually a 24-hour or timed urine specimen is collected, and a blood specimen is also collected from the patient, and then the comparison of the urine specimen in that component and the blood specimen of that component is created, and then the clearance is calculated. So this is all about the renal function test. Hope I am able to clarify you all the concepts of the renal function test. Students, if you have any query related to that session, you may write in the feedback section on your LMS, which is there on your LMS only.